Exocytosis is the reverse process of moving material into a cell. Kumbaga, siya yung kabaliktaran ng endocytosis na diniscuss nung previous na group. Exocytosis is a process by which a cell transports secretory products through the cytoplasm to the plasma membrane. Secretory products are packaged into vesicles. Vesicles are membrane-bound spheres. Ang definition ng exocytosis. Ang exocytosis, it describes the process of vesicles fusing with the plasma membrane and releasing their contents to the outside of the cell. Endocytosis is the process of capturing a substance or a particle from outside the cell by engulfing it with the cell membrane and bringing it into the cell. Kumbaga, para pasimplihin yun, kung ano yung in-explain ng endocytosis, di ba? Kumuha sila na parang substance mula sa taas, tapos dumaan sa cell membrane, binalot, naging vesicle sa loob. Ngayon, yung exocytosis, kabaliktaran naman yun. Bali mula sa bilog vesicle sa loob, yung waste na nasa loob nun, parang iaakyat, mapupunta sa plasma membrane, tapos mapupunta sa labas ng cell. Why is exocytosis important? It is important because it allows cells to secrete waste and substances molecules such as hormones and proteins. It is also important for chemical signal messaging and cell-to-cell communication. So, importante siya mainly because para siyang naglalabas ka lang na kumbaga parang dumi mula sa cell. Ini-secrete mo siya. So, ayun. So, what cell uses exocytosis? Exocytosis is used by animals and plant cells to secrete waste from the cells. In exocytosis, vesicles containing substances fused with the plasma membrane, the contents are then released to the exterior of the cells. So parang ayun nga, parang baliktad lang ng endocytosis. Meron 3 types of exocytosis. Constitutive exocytosis, regulated exocytosis, tsaka lysosome-mediated. Constitutive exocytosis, it involves the regular secretion of molecules. This action is performed by all cells. Constitutive exocytosis functions to deliver membrane proteins and lipids to the cell surface and to expel substances to the cell's exterior. So nagagamit ang constitutive exocytosis ay magagamit ng all forms of cells, kumbaga kahit anong cell pa yan. Sa regulated exocytosis naman, relies on the presence of extracellular signals from the expulsion of materials within vesicles. Regulated exocytosis occurs commonly in secretory cells and not in all cell types. Secretory cells store products such as hormones, neurotransmitters, and digestive enzymes that are released only when triggered by extracellular signals from cytoplasm. So sa regulated exocytosis, hindi katulad ng constitutive, sa regulated medyo siya, kumbaga mas sensitive kasi hindi lahat ng cells kayang i-execute yung regulated exocytosis. Kumbaga, dapat ang nakakapaggawa lang ng regulated exocytosis ay yung mga secretory cells, kumbaga. Sa lysosome-mediated naman, these organelles contain acid hydrolase enzymes that breaks down waste materials, microbes, and cellular debris. Lysosomes carry their digestive materials to the cell membrane where they fuse with the membrane and release their contents into the extracellular matrix. So parang sa lysosome-mediated, parang especially siya for lysosome lang. Doon naman tayo sa steps of exocytosis. Ang steps of exocytosis ay trafficking, tendering, docking, priming, and fusion. First, trafficking. Vesicles are transported to the cell membrane along with the microtubules of the cytoskeleton. Movement of the vesicles is powered by the motor proteins, kinesins, dyneins, and the myosins. Sa tendering naman, upon reaching the cell membrane, the vesicles becomes linked to and pulled into contact with the cell membrane. Sa docking involves the attachment of the vesicle membrane with the cell membrane. The phospholipid bilayers of the vesicle membrane and cell membrane begin to merge. Sa priming occurs in regulated exocytosis and not in constitutive exocytosis. This step involves specific modification that must happen in a certain cell membrane molecules for exocytosis to occur. So, sa priming, ano lang siya, parang specialized siya for regulated exocytosis lang, hindi siya pwede sa constitutive exocytosis.
Yung huli, fusion. Incomplete fusion, the vesicle membrane fully fuses with the cell membrane. The energy required to separate and fuse the lipid membranes come from ATP. Kiss and run fusion, the vesicle temporarily fuses with the cell membrane long enough to create a fusion pore and release its content to the exterior of the cell. The vesicle then pulls away from the cell membrane and reforms before returning into the interior cell. Kumbaga sa fusion, dito na nangyayari yung nagsasama na yung vesicle pati yung cell membrane para makabuo sila ng pore, para mailabas nila yung waste material palabas ng cell. So, yun lang yung buong exocytosis. Bye-bye!